Today, we've got a fascinating story for you from the history of medicine. It's about a wild experiment that not only launched an entire field of therapy, but also created a really strange paradox, a mystery that gets right to the heart of how science actually works. So, let's dive in.
And here is that paradox. The man who's literally called the father of modern hyperbaric medicine, Dr. Boerema. He published this one paper that's considered the absolute foundation of his field. But if you look at modern research, you'll almost never see it mentioned. How does that even happen? How can the most important paper be the one nobody seems to cite? Well, to solve this little mystery, we have to go back and understand the incredible experiment that started it all.
So, let's set the scene. It's 1960. A team of researchers in Amsterdam, led by Dr. Boerema, decided to ask a question that sounded, well, completely nuts. Could an animal actually live without blood? Or, to be more specific, without the red blood cells that are supposed to carry all the oxygen?
Their plan was audacious, to say the least. First, they took some piglets and did something called an exchange transfusion. Basically, they washed out almost all of the piglets' red blood cells and replaced them with a simple plasma solution. Then, and get this, they put the piglets inside a special high-pressure chamber and just flooded it with pure oxygen at 3 times the normal pressure of the air we're breathing right now. And after all that, they just watched to see what would happen.
Okay, and this is where the story goes from wild to almost unbelievable. After they did all that, the hemoglobin level in the piglet's blood, the protein that carries oxygen, it dropped to less than 0.4 grams per deciliter. To put that in perspective, a healthy piglet should be somewhere around 10 or 12.
Let me be absolutely clear here. By every medical rulebook then and even now, that number is considered fatal. It's just not compatible with life. Without hemoglobin to shuttle oxygen around, those piglets should have died within moments. But they didn't.
The piglets lived. Now, this wasn't some kind of magic trick. It was physics. Boerema had figured out a way to pull an end run around one of the body's most basic rules. And in doing so, he proved that this whole idea, hyperbaric medicine, was for real.
So here was the secret. Under that intense pressure, oxygen doesn't need to hitch a ride on hemoglobin anymore. The pressure itself literally forces oxygen molecules to dissolve directly into the liquid part of the blood, the plasma, and it dissolves in big enough quantities to keep all the body's tissues alive and well. He proved the plasma itself could do the job.
And what he demonstrated beautifully was a basic law of physics called Henry's law. You've actually seen this law in action every single time you've opened a can of soda and heard that fizz. It's the exact same principle.
Yep, it's really that simple. You crank up the pressure of a gas sitting on top of a liquid, and more of that gas is going to dissolve into the liquid. Boerema's experiment was the first time anyone had proven this law works so perfectly in a complex living animal. It was a complete game changer.
But here's the thing. This wasn't just some cool lab trick destined for a textbook. The implications were huge, and they turned into real-world medicine with, well, incredible speed.
Just look at this timeline. The pace is just stunning. The team publishes their initial research in '59. Then comes the big one, the Life Without Blood paper we're talking about, in 1960. And by 1961, just one single year later, this discovery is already being used to save human lives.
The first huge clinical win came from one of Boerema's colleagues, W.H. Brummelkamp. He used this new technique, which we now call Hyperbaric Oxygen Therapy, or HBOT, to treat gas gangrene. This is a terrifying bacterial infection that absolutely thrives where there's no oxygen. So by putting patients in a hyperbaric chamber, he could just drench their infected tissues with so much oxygen that the bacteria, well, they couldn't survive. It was the first undeniable life-saving use for HBOT.
All right, so now we've seen the experiment and its incredible impact. We have all the pieces of the puzzle. So let's circle back to our original question. If this paper was so revolutionary, why doesn't anybody cite it anymore?
Well, the answer really comes down to understanding that there are 2 different kinds of important papers in science. On one side, you've got the seminal paper. That's Boerema's. It's the scientific bedrock. It's the one that proves a radical new idea is even possible. But on the other side, you have the most referenced paper. That's usually something like a big modern review that pulls together all the latest evidence and basically tells doctors how to use a treatment today.
And right there, that's the key. Think about it. A doctor today who's treating, say, a diabetic foot ulcer isn't going to cite a piglet experiment from 1960 in their notes. No, they're going to cite the latest big review that summarizes all the human trials. Boerema's paper is the origin story, the deep foundation. But that modern review, that's the practical, up-to-date user manual. One is the reason the field exists at all, the other is how the field gets practiced day to day.
So the mystery, it kind of dissolves, doesn't it? It's really just a lesson in how science moves forward. Boerema's work is so fundamental, so foundational, that it's just become assumed knowledge. It's a foundation buried so deep we don't even think to look at it anymore. Which kind of leaves us with one last thought. What are the foundational, rarely cited ideas that your world is built on? Your job, your hobbies? Chances are, they're everywhere, quietly making everything else possible.